Hello everyone, my name is Shivansh Gaur and I welcome you to my channel Biological. Today we are going to talk about on a topic blood groups. This topic is very important and this topic is one topic in which a lot of students get confused. So today I am going to tell you what are the different blood groups and which blood group system do we study. And before moving on towards our today's topic, I would also like to tell you that now I am a plus educator on Unacademy. And I take biology classes in the EAMCET category. So in the AP and TS EAMCET, I teach biology. So now you can also find my video lectures on the Unacademy. You go to Unacademy app, download the Unacademy learning app right now, and I will now tell you how to take the different classes, the special classes, the crash courses and the plus subscription. So if you're not there at the Unacademy Plus, you should definitely download the Unacademy app, learning app right now and become the Unacademy Plus subscriber. What the benefits will you get? You will get the benefit of the premium content at a very affordable price. You will get structured live courses and dedicated live doubt clearing sessions. You will get quizzes at regular intervals for self-analysis, and you will also get the different batches for the complete preparation in all the subjects, whether it is physics, chemistry, maths or physics, chemistry, bio. And there will be test series or DPPs, full length mock test, etc. with analysis, and this single subscription will give you this unlimited access. You will be getting all of this in just single subscription. Okay, there are more than 20 plus educators, 20 plus top educators there. There are 10 plus batches already running, 250 plus courses, 2000 plus lessons, and many more are added every day. You just use the code LIVEShivansh and you will get each and everything in just one subscription. So go for the Unacademy Plus subscription right now, because these batches, they are there already running for 2021 aspirants. You can go for the 12-month plan, which is 14,850, and you can also go for the 6-month plan. Use my code LIVEShivansh. You use my code LIVEShivansh and this code will give you additional 10% discount. So what else you can ask for? Go to the Unacademy learning app, take the subscription, use this code LIVEShivansh. When you're buying the subscription, you will get additional 10% discount. See the batches that are going on: comprehensive batch for PCM and comprehensive batch for 2021 PCB. There are also crash course batches for the aspirants, for the 2020 aspirants. There are crash course batches in which I am also there teaching with my biology crash course there. Okay, so the crash courses are there, with the rank booster batches, they are there. So enroll now. So if you want to learn from me and the other top educators, then download the Unacademy learning app right now and follow me there, and also take the Unacademy Plus subscription for the descriptive and detailed courses. Now moving on towards our today's topic. The today's topic is blood group system. So now today we are going to do the blood groups. And the blood groups that we have in syllabus that we talk about is the ABO blood grouping system. So now I am going to tell you how are the different blood groups that are decided. What is this ABO blood grouping system? How are the different individuals, they have different blood groups, and why do we need to match the blood group before we do the blood transfusion? So we are going to talk about this particular topic in very much detail. So now starting with this, we have 4 types of blood groups. In ABO blood group system, we have 4 types of blood groups. What are the 4 types of blood groups? See, the 4 types of blood groups are A, B, AB and O. So these are the 4 types of blood groups. And how are these blood groups decided? How is that decided that a particular person has a blood group of A or a particular person has a blood group of B? How is that decided? Now see, this blood group, whether it is A, B, AB or O, it depends upon the antigen, the specific antigen which is present on the surface of RBC. So how is that this blood group decided? It is decided by the antigen which is present on the surface of RBC. Red blood cells, as you all know, RBCs, they are the formed elements. They are one of the formed elements in the blood. Okay, so now this antigen, it is present on the surface of RBC, and the RBCs, they have different antigens which are present there. So now if the antigen present is A, for example, if on the surface of RBC the antigen which is present is A, then the blood group will be A. Now if the antigen which is present on the surface of RBC is B, then the blood group will be B. If the antigen present on the surface of RBC is both A and B, then the blood group will be AB. Now what happens is, we'll also talk about the genetics of this blood group. In a different video. In a different video, I'll make a different video in which I will tell you that the genes which are coding for these antigens, these polypeptides, these are codominant genes. They are codominant genes. So which means that they both express when they are present together. So that is how both the antigens, they are present on the surface of RBCs. So blood group becomes AB. Now, if there is no antigen, that is also the case. If there is no antigen on the surface of RBC, then the blood group becomes O, which means there is no antigen on the surface of RBCs. Now, what are antigens? The question arises, what are antigens? So, antigens, what are antigens? Antigens, they are the chemicals which induce chemicals which induce the immune response. The chemicals which induce the immune response. We all know this. Okay. So these are the chemicals, and these chemicals, they are present on the surface of RBC. Keep that in mind, that what are antigens? These are the chemicals which induce immune response. And in response to that antigens, to these antigens, what are produced? Antibodies are produced. What are produced? Antibodies are produced. And these antibodies, these are basically protein molecules. These are protein molecules produced in response to antigens. Now keep these two definitions in mind, okay, because now what next I'm going to tell you, it's going to help you. These definitions are going to help you. So I'm telling you that keep these definitions in mind. What are antigens? These are the chemicals which induce immune response. And what are antibodies? These are protein molecules which are produced in response to these antigens. Now, as I already told you that these blood groups, they are decided by the presence of a specific antigen on the surface of RBC. Now, keeping that thing in mind, we will suppose and take 4 cases. So let's do that. You'll be able to understand that in a very simple way. Let us take 4 individuals. And give them 1 blood groups. Okay. So now see, I'll draw 4 individuals. So this is individual 1. This is individual 1. This is individual 2. Okay. This is individual 2. This is individual 3. And this is individual 4. So these are the 4 individuals. Okay. And let us suppose that this is the RBC of individual 1. This is the RBC of individual 2. This is the RBC of individual 3, and this is the RBC of individual 4. So we have 4 individuals, 1, 2, 3 and 4. So these are the 4 individuals that we have. And now since we know that these are the RBCs which are present in his blood, okay, and let us suppose that the individual one has the blood group of A, okay, so what will be the antigen which will be present on the surface of its RBC? It will be antigen A. Very simple, okay. Antigen A will be present on its surface, on the surface of his or her RBCs, and that is why his or her blood group is A. For individual 2, let us suppose blood group is B, so the antigen will be present will be B. Very simple, okay. Now the individual 3, let us suppose, has an blood group of AB, so the antigen will be which will be present on its surface will be AB, and in another case, like the 4th case, let us suppose that this individual doesn't have any antigen on its surface, so its blood group will be what? O. Its blood group will be O, and there will be no antigen on the surface of RBC, so there are no antigens on the surface of RBCs, okay. Now as I already told you that these antigens are present on the surface of RBCs. Now there are antibodies also which are present. And these antibodies, they are not present on RBCs, they are present in the plasma. So let us suppose that if this is RBC and this, this whole square, this whole rectangle or whatever I have made, it becomes plasma. So let us suppose this is the plasma. Okay, this is plasma, the liquid portion of the blood, the fluid part of the blood, okay. And see here, if this individual has antigen A on the surface of its RBC, antibody which will be present will be B. It cannot be A. Why it cannot be A? Because as I already told you that this antibody, it fight against these antigens. They are produced in response to the antigens. So if this was antibody A, then this antibody A would have killed these RBCs, and destroyed these RBCs. So there cannot be antibody A. It has antibody B. Likewise, in this case, since antigen present is B, there cannot be antibody B. The antibody which is present here is A. Because if antibody B is present here, it will, the plasma of this individual or the plasma of this individual will kill its own RBCs. It will destroy its own RBCs. So that cannot happen, okay. Now in this case, see, since both the antigens are present, antigen A is also present, antigen B is also present. So since both the antigens are present, there cannot be any antibody. See, if there is antibody A, then too it's going to destroy this, this RBCs, because there is antigen A. If there is antibody B, then too it's going to destroy the RBCs because there is antigen B. Okay, so there cannot be any antibody. So here the antibody, they are nil. Here the antibody is nil. There is no antibody in the individual of blood group AB. That's very clear. Now we have the 4th case. The 4th case is that it has no antigen. So since this individual has no antigen, it can have both the antibodies. Because there is no antigen. So if both the antibodies are there, of course there is no antigen. So these antibodies are not going to react with this particular RBCs. So there is no problem. Okay. So now I think till now everything is clear. These are 4 individuals with 4 different blood groups having antigens on the surface of their RBCs and antibodies in their plasma. Now, as I told you, before the blood transfusion is done, the blood group is matched. Why is it? Why the blood group needs to be matched? Okay, see, very simple. This individual has blood group A, the antigen present is A. Now, if suppose the blood group matching is not done, and by chance if this individual receives the blood group from individual which has blood group B, now when this blood reaches this blood, when this blood is there transfused into the individual A, what will happen? The antibodies A will start killing the RBCs, because this is antigen A. And as I already told you that these antibodies are produced in response to the antigens, and they fight against the antigens. Or they kill the antigens. So what they do, this antibody will start killing the RBCs of this individual, and the blood transfusion will not be successful. Likewise, this case, if B is transfused the blood group A, then the same thing will happen, because it has antibodies B, so it will start killing the antigens of this individual, and it will destroy the RBCs of this individual. So likewise, we have to done the blood group matching. So this blood transfusion is not possible. This A can receive the blood from A, that is there. A can receive the blood group blood from A. B can receive the blood from B, but A cannot receive blood from A. B cannot receive blood from A, and A cannot receive blood from B, vice versa. Okay, now here pay very close attention. It has both the antigens. But it has no antibodies. So there is no one to fight against the antigens. Got my point? Since it has no antibodies, it has no, since there is no antibody, so it has no tendency. There is no tendency to fight against any antigen, because there is no antibody. Antibody is nil. So even if we give blood of A, it will receive the blood of A, because there is no antibody to fight against the antigen. So very simple. Even if we give the blood of A, it will receive the blood of A. Even if we give the blood of B, then too it will receive the blood of B, because there are no antibodies to fight against the antigen. Of course there is antigen coming, but there is no antibody to fight against it, so it will receive the blood group from A as well, from B as well, and AB. Of course AB can give blood to AB, same blood group can be transfused. That is very easy. Now see the fourth case. In the fourth case as well, it can give blood to AB, because again there is no antibody to fight against any antigen. There is no antigen as well. In this there is no antigen as well, so it can receive the blood from O as well. So that is how AB becomes universal. What do we say? Universal acceptor. Got my point? How does AB become universal acceptor? Because it has no antibodies. It has nil antibodies, and therefore it can receive blood from anyone. Now see here, pay very close attention. It doesn't have any antigens. It doesn't have any antigens. Okay, but it has both the antibodies. This is the problem. It has both the antibodies. It has no antigens. So now what happens is, even if we give blood, the blood group of O, even if he gives his blood to AB, since there is no antigen, so there will be no response of the antibody, since it has no antigens. Okay, now even if the blood group of O person, this person, gives blood to B, now what happens? Now B has antibodies of A, but it has no antigen. So with this antibody will fight against whom? There is no one to fight. Okay, if this individual gives blood group to A, like blood is transfused to A, then too, since A has antibodies B, but there is no antigen, again it can give blood to A. So that is how O becomes universal donor. A becomes universal donor. I hope this has been made very easy for all of you, and you can understand how AB become universal acceptor and O become universal donor. So O can receive blood from only and only O. Why? Because A can receive blood from A and from the universal donor. B can receive blood from B and the universal donor. AB can receive blood from anyone. O can give blood to anyone, but it can receive blood from only and only O, because it has both the antibodies. Both the antibodies. Got my point? Got my point? So this is how we define the ABO blood group system. So these are the 4 blood groups that we have talked about. In the next video, I will be talking about the RH factor. RH factor that will be dealt in the next video. Next video will be on RH factor, RH grouping. That is also another kind of antigen which is present on the surface of RBC. And you may have heard about this, like A positive, B negative, O negative. So where is this positive and negative coming from? I'll be telling this in my next video. I hope ABO blood group system is clear to all of you. Till then, take care. I will meet you in the next video. Bye bye.